Hello everyone. In this video, we will discuss about the non-cellular organism which includes viroids and prion. So first, what is meant by viroids? So viroids are the small particles which are smaller than viruses, which is made of which is the short pieces of naked RNA only 300 to 400 nucleotides long with no protein core. Viroids were discovered and given their name by American plant pathologist Theodore Diener in 1971. They are the smallest infectious particle which are capable to cause a disease in plants, animals, and humans also. The nucleotides are often internally paired, so the molecule has close folded three-dimensional structure that presumably help protect it from attack by cellular enzymes. The RNA does not code for any proteins or protein is absent in viroids. Remember, protein particles are absent in viroids. Thus, for viroids having conclusively identified pathogen only of plants, but it can be caused disease in human and animal also. Viroids cause some plant disease such as a potato spindle tuber disease, which is first time identified by the scientist. Next one is the prion. Prion is the few infectious disease are caused by the prion. So what is meant by prions? Prions is the protein particle. In 1982, American neurobiologist Stanley Prusner proposed that infectious protein cause a neurological disease in sheep that is called as a scrapie. The infectivity of scrapie infected brain tissue is reduced by treatment with protease but not by treatment with radiation, and this suggesting that the infectious agent is a pure protein. Prusner coined the name prion for a proteinous infectious particle. There are 9 animal diseases now fall into this category, including the mad cow disease that emerged in cattle, which is identified in Britain in 1987. The human disease that are Kuru, then Jacob disease that is a CZD are the disease which is caused by the prions. These diseases are caused by the conversion of normal host glycoprotein, which is called as a capital P small r PC for the PRPC for cellular prion proteins into an infectious form that is called as a PRPSC. S for the scrapie protein. The gene for the PRPC is located on chromosome 20 in human. In case of human, when fragments of PRPSC molecule accumulate in the brain forming the plaque, these plaques are used for the diagnosis, but they do not appear to be cause of cell damage. This is about the prions. So, what is meant by prions? The prions is a proteinous infectious particle. The protein which is responsible to cause the disease is the PRPC. When this PRPC, that is the cellular prion protein, when it after the infection, it get converted into PRPSC form, and this PRPSC form is responsible to cause the disease. So next is what is the major difference between viroids and prions and virons. Let me discuss about the difference. For the here there is a structure of the normal prion protein, that is the PRPC, And here is the infectious or abnormal prion protein, that is the PRPSC, which is responsible to cause the disease. Next is the difference between virus, viroids and prion. First one is the about the genome. In case of viruses, there is the maybe DNA or RNA. It may be uh, double stranded or single stranded. In case of viroids, the genetic material is in the form of RNA and it is always the single stranded. And in case of prions, genetic material, that is the DNA and RNA, is absent. Okay, but protein is present, and this protein is the single stranded material. Next one is the coat or envelope. So in case of viruses, the coat is made up of capsid and envelope. Or in case of viroids, the coat is made up of for HDV only. HDV is the hepatitis D virus. And in case of prions, the coat is absent. Next one is the target or host. So in case of virus, they are capable to cause the disease in case of animal, plant, bacteria and RT. In case of viroids, they are capable of to cause the disease in plants, but there is a research that they may cause the disease to animals and humans also. And next one is the prions, which are capable to cause the disease to animals. Next one is the host cells or organ. So viruses show all types of cells as a host. Viroids used only plant cells as a host, and the prions, they use the nervous system of the animal as a host. Next is the mode of transmission. So in case of virus, they may cause the disease by air, water. Next is the viroids. So viroids can before discussing about the viruses can be spread through air, water, then uh, droplets, blood, etc. The recent example is the coronavirus, which is spread by the air or droplets. Next is the viroids. The viroids infection are transmitted by cross contamination following the mechanical mechanical damage to plants as a result of horticulture or agriculture practices. Some are transmitted by aphids. They can also transfer from plants to plant by leaf contact. Next is the mode of transmission for the prions. The prions are the organized protein. So they are spread by three different ways, which include acquired, familiar, and sporadic transmission. Current research suggests that primary method of infection in animal is through ingestion. Okay, this is all about the viroids and prions difference. One major last difference is in between viroids and prion is that viroids are sensitive to RNase but resistant to proteinase. Why? Because in viroids RNA is present and protein coat is absent. In opposite, in case of prions, so prions are sensitive to protease enzyme but resistant to RNase enzyme. Why? Because in case of prions, there is a protein coat is present, so they are sensitive to protease enzyme, but RNA or DNA is absent, so they are resistant to RNase enzyme. Okay, this is all about the viroids and prion. So expected question on this topic are the definitions of viroids and prions and difference between viroids and prions, or write a short note on viroids, or write the short notes on prions for 4 marks. Okay, thank you.